Now, I told you yesterday when we were discussing the bacteriophage and the different parts of the bacteriophage that we would discuss the life cycle of a virus using the bacteriophage. So, here we go with our life cycle. So, on your line there where it says number 1, you should write what is on this slide right here. The virus attaches to the cell using tail fibers to adhere to the host cell's membrane. So on this diagram here, as I am looking, this little, I don't know what we're going to call this, a light orange with a darker orange on the membrane, this is the host cell. And as you can see, inside the host cell, we have a maroon plasmid of DNA. A maroon plasmid of DNA. So, match your picture for this one where the cell is just attaching itself and put that on number 1. And then your writing definition of number 1 down below that should be the virus attaches to the cell using tail fibers. Okay? Now the second step in the process is where the virus injects its genetic material into the cell by adhering the base plate and releasing DNA from the capsid through the tail sheath. Remember we said yesterday that that tail sheath injects, it acts like a syringe and it injects the DNA from the capsid into the host cell. So as you can see in the diagram I have up there, the DNA that was up here in the capsid traveled down through the sheath and is now being injected into the host cell. We can see this because remember, the host cell DNA was maroon. Now the DNA that we are putting in there, that the virus is putting in there, the DNA that the virus is putting in there is blue. And once again, I guess maybe I shouldn't say DNA because I should use genetic material. It could be RNA, just remember that. Okay, so I'll try to use genetic material instead of just DNA. But the fact is that the second thing, after the virus adheres itself to the host cell, it has to place its DNA, its genetic material, into that host cell. The next step in the life cycle of a virus is that the cell reads the virus DNA. And in that process, the cell is instructed to make more virus parts. Okay? More virus parts. So I can see, looking at this particular picture, okay, I can see that we have, these things in here. We have tail fibers right there. Here's genetic material. Here is the sheath, the tail sheath. Here's the capsid, and it's also got genetic material. Maybe that is a base plate right there. But the whole idea is that the DNA from the virus now instructs that cell to make DNA parts, to make, virus parts. Whether it's the capsid here, the genetic material, the sheath, the base plate, and the tail fibers. Okay, we are making all the parts for the virus. So what do you think is going to be our next step? What's the logical step? After we've made the parts, we're going to put them together. Excuse me. So, number 4 there, what we should write for number 4 is that the virus parts are assembled. We've got all the parts. You know, we see the head. We see now the genetic material. Boom, there's the capsid. We see the base plate, then the sheath. There we go. And finally, the tail fibers putting together. So now, as you can see here, in our cell, in our host cell, we have assembled the virus. And in your host cell, you should see many. Your picture has many different viruses that are assembled in it. More so than what my picture does up here. Okay? Now, the virus is ready to go. Once we've assembled the virus, it's as good as it's going to get. Remember, one thing that viruses do not do, well, a couple of things. Who can explain to me? Remember from yesterday, what are some things that viruses do not do? They don't grow. Okay, what else don't they do? They don't reproduce without a host. What do they do? They mutate. They do mutate. But they don't reproduce without a host. They don't grow. So this virus right here is as good as it's going to get. So what happens next, the final step, is that the new viruses have to emerge from the cell. So basically, they keep making viruses, keep making viruses, keep making viruses, keep making viruses. Okay? It's like filling a balloon with air. Eventually, what's going to happen? It's going to pop. We call that it's going to lyse. L-Y-S-T. When it lyse, it bursts. And when it bursts, look what's happening. What happens? The cell is destroyed now. The membrane is gone. That cell is no longer any good. And what else happened? All the viruses are out moving around now. What do you think's going to happen? It's going to start number 1 all over again. It's going to attach to another cell. Okay? Then it's going to inject the DNA. Then the DNA is going to cause that cell to build new parts. And then they're going to assemble the new parts. And then finally, boom, it's going to burst again. This continuous cycle, the continuous cycle that is happening. Okay? So, I guess, theoretically, okay, what do we do? How do we get rid of a virus? Yeah, we go to the doctor and get some meds, right? Remember I told you that some of these meds are what causes the mutations. Okay, we could get some medicine. We take the medicine, all of a sudden the medicine kills all of the virus, and we're fine, we're good to go again. Okay? But do you understand now the life cycle that these viruses take? Okay? Could you repeat it? I hope you can, because as you can see tomorrow, we're going to take our test in here. Okay?